Hello everyone, I'm Yang Liu. It's my pleasure to introduce our recently published research in iMeta, titled Bacterial Social Interactions in Synthetic Bacillus Consortia Enhance Plant Growth. This study explores how social interactions within synthetic Bacillus consortia influence plant growth and proposes a framework for the rational design of microbial communities. The plant rhizosphere microbiome, often called the second genome of plants, plays a crucial role in nutrient uptake and plant health. Plant growth-promoting rhizobacteria, PGPR, are widely used, but their effectiveness is often influenced by complex microbial interactions. This study aims to unravel social action mechanisms among Bacillus strains and incorporate them into the design of synthetic consortia. Bacillus strains display social behaviors such as swarming and kin recognition, where kin strains tend to cooperate and non-kin strains often antagonize each other. We aim to link phylogenetic relatedness with social behavior and plant growthing traits to construct more efficient consortia. This study features 4 key findings. First, inoculation with PGPR strain Bacillus velezensis SQR9 promoted cucumber growth and enhanced cooperation among kin strains in the rhizosphere. Second, strains with higher phylogenetic relatedness exhibited more overlapping social behavior and metabolic niches, to stronger synergistic effects. Third, consortia with moderate relatedness showed the most effective plant growth promotion. Our results offer a new perspective for designing effective microbial consortia. Now let's walk through the methodology. We first performed pot experiments by inoculating SQR9 and conducted 16S rRNA and GYRA gene amplicon sequencing to assess changes in the rhizosphere bacterial community, especially Bacillus populations. We then isolated rhizosphere Bacillus strains and evaluated their social interaction phenotypes, carbon source utilization patterns, and plant growth promoting traits to explore links between phylogenetic relatedness and functional traits. Based on these results, we constructed 30 Bacillus consortia of either high or moderate relatedness across 4 richness levels and tested them in hydroponic and natural soil systems to evaluate plant growth effects. Lastly, we scaled up to 300 consortia for expanded validation. Here are the key findings. Inoculation with SQR9 significantly altered the composition of the cucumber rhizosphere bacterial community. Notably, the abundance and structure of Bacillus populations changed substantially, suggesting that Bacillus plays a central role in rhizomicrobiome modulation. We isolated Bacillus and closely related strains from the rhizosphere of inoculated and non-inoculated plants and performed pairwise swarming confrontation assays. SQR9 altered the distribution of swarming phenotypes among rhizosphere Bacillus strains, steering the community toward higher fusion and lower antagonism. This behavior is regulated by a mechanism called kin discrimination, highlighting the role of phylogenetic identity in shaping bacterial social interactions. Next, we examined the relationship between phylogenetic relatedness and carbon utilization patterns among Bacillus strains, finding that more closely related strains shared more similar carbon profiles. By integrating phylogeny, social interaction behavior, metabolic traits, and growth-promoting potential, we constructed consortia across 2 levels of relatedness and 4 richness levels. Plant growth experiments showed that moderately related consortia outperformed highly related ones in increasing cucumber height, dry weight, root colonization, and in producing IAA and siderophores. Thus, moderate relatedness promotes optimal consortium performance. We further constructed 300 Bacillus consortia to validate the findings at scale. The results again confirmed that moderately related consortia exhibited greater stability and stronger plant growth promoting capacity. To conclude, this study is the first to show that PGPR strain SQR9 can direct rhizosphere community succession, especially within Bacillus and its relatives, toward a more cooperative structure. Leveraging this ecological shift, we constructed moderately related Bacillus consortia and validated their superior performance in both hydroponic and soil systems. Our work introduces a novel ecological framework incorporating social interaction and carbon niche competition into synthetic consortium design, offering new strategies for smart Bacillus community assembly and sustainable bioinoculant development.